I covered quite a bit of material yesterday, and so I want to go through and finish up talking about structures of carbohydrates, and then we're going to get into some fun stuff. And a lot of people look at metabolism and think it's really not very fun, but I hope I'm going to convince you with metabolism that you understand how your body works. And it takes a little while to get into that, but once you get into it, it's like, oh yeah, now it makes sense. So hopefully that'll happen for you as well. Before I get there, though, I need to finish up a couple things. I finished yesterday mentioning amino sugars. And amino sugars, as their name suggests, are sugars that have an amine group attached to them. As I noted yesterday, that amine group is usually attached at position 2. There may be other things that are attached as well. The reason I show you the one on the left is it's a monomer of a polymer that we oftentimes find in nature, and that is a polymer known as chitin. And chitin is the material that's the exoskeleton of insects. And it's a polymer of N-acetyl-glucosamine, this guy that you see right here. Okay? So there are polymers of things besides glucose that are out there that make sense. Okay. And chitin looks like, there you go. There's the linkages. You don't need to worry about the linkages, but just know that it's a polymer of that. Another interesting polymer are those of peptidoglycans. And so I need to say a little bit about those. So the name peptidoglycan tells you a little bit about their structure. Peptido telling you that it's a peptide, meaning that there are some amino acids in there and they have peptide bonds between them. And as we can see here, the cross-links that exist between these guys are actually peptides. So you see these two tetrapeptide chains cross-linked by a peptide. These are short peptides that join these guys together into sheets. And these make up the cell walls of bacteria. Okay? So one of the ways that bacteria have a relatively rigid cell wall is because they have a peptidoglycan layer. And I've actually briefly mentioned this layer to you at the very beginning of class when I talked about unusual amino acids. So I want to say just a word about this because this is kind of interesting. I talk about how in biology we have these various defenses that organisms have evolved over the years to protect them. And so for a bacterial cell, the life out in the real world is pretty hazardous. There's all kinds of things that can kill that bacterial cell, not the least of which are proteases. We've seen some of the proteases already. And the proteases are found in places not just in the digestive system, but you have proteases in your lungs, for example. We'll talk about those in a little bit. And proteases in your eyes and so forth that help provide protection because they can break down proteins in bacterial membranes. Well, many bacteria have evolved a protective mechanism in their peptidoglycan layer in that they have an unusual amino acid that they put into this peptide. And the unusual amino acid that they have is known as d-alanine. Okay? You may remember when I talked about the amino acids to begin with, I said that virtually all the amino acids that are produced biologically on the face of the earth are in the L configuration. Well, by putting an amino acid into the D configuration, it basically makes it undigestible by a protease. So these peptidoglycan layers are largely protected from degradation by proteases, which are aimed at breaking down all proteins that have L amino acids within them. So that's an evolutionary, interesting sidelight that pops up in biology. It's one of the very few places in all of nature we find a d-amino acid. Okay. And I also want to mention proteoglycans. So proteoglycans are sort of related, although I would say not especially related. But proteoglycans are complex molecules that look kind of like this guy. And they have a sort of a backbone of hyaluronic acid. Now, hyaluronic acid is a polymer. It's a very long polymer of a couple of modified sugars. And the sugars are modified to have negative charges on them. So most sugars don't have a charge on them. They have OH groups and so forth. But these sugars have been modified to have negative charges on them. And more importantly, some of these other, and this is a polymer of these. Okay, so it's a polymer of these negative charges. So too are chondroitin sulfate and keratin sulfate. These guys are polymers of monomeric sugars that have negative charges. So the message I'm trying to convey to you here is that this is a structure that's held together partly by hyaluronic acid, partly by a protein, all right? And these things make very feathery kinds of, I don't want to say displays, but very feathery kinds of structures. And the reason that they're very feathery is because of all of the negative charges that are on here, okay? So if we think about this as a polymer, and these guys are all polymers, and every one of those is loaded with negative charges, we think about what negative charges do with each other. They're going to repel each other. And so what happens with these guys is they, when they're put in aqueous solution, that is in a water environment, when they're put in water, which is how we find them in nature, they spread out. They spread out to get as far, each strand trying to get as far away from the other ones as it can, because it's fully negative and all these other guys are fully negative as well. Well, what does that mean chemically? Well, chemically what it means is that, I shouldn't say chemically, but physically what it means is that these guys have they give water a very unusual property. And the unusual property that they give to water is they make it very slimy. Very slimy, okay? So snot, for example, is full of this stuff, all right? And it's slimy because these groups are all out there trying to avoid each other, and as a consequence, they really organize the water around them in a very unusual fashion. So water doesn't behave like water, it behaves more like a lubricant, which is what these things actually do. Hyaluronic acid is a important component of synovial fluid. So synovial fluid is literally the lubricant that's in your joints. When people get old and their joints don't have enough synovial fluid, they will complain about, oh, wow, it hurts to walk, it hurts to do all this sort of stuff. It happens because they don't have enough lubricant between their bones or their cartilage that's interacting. So in your knees, for example, you're actually cushioned every time you take a step by this synovial fluid that keeps that cartilage and those tissues from hitting each other, okay? And again, it has this very slimy, very lubricating property like I've described to you here because of the structures that you see on the screen, okay? So that's an important consideration, and the older you get, the more you will appreciate your synovial fluid. There's your lesson for the day. Okay, the last thing I mentioned, I've mentioned these before, these are the blood groups. So blood cells have on their surface, they have what are called antigens, and those antigens differ in their configuration. Now, I'm not asking you to memorize which one is which or any of this, but it shows some of the different displays that can be on there. So what these are glycoproteins, that is a protein that's embedded in a membrane, and onto it, it has several sugars or modified sugars attached to it. So here's L-fucose attached to these two guys, attached to this guy up here. That's the type A blood group. Here's a type B blood group seen over here. And you see there's a lot of similarity, but there's also difference in terms of what's attached out here. Now, the reason we call them antigens is because they are immunogenic. And if you transfer the wrong blood type into a person, that person's immune system will attack it as a foreign invader and cause a problem. So you want to match blood types as much as you can to do that. One of the things about type O blood, I'm a type O blood myself, is that O blood is known as the universal donor, and the universal donor doesn't have these groups on the end at all and doesn't stimulate an immune response. And that's why they are what they are. Okay, so identity happens, I mentioned this before, as a result of the configuration of these sugars that are on the surface of these blood cells, just like we have other configurations on the surface of kidney cells or liver cells or whatever. Okay. So that's the last of what I want to say with respect to structures of carbohydrates, unless there are questions. And if not, I will move forward. Yes?
What does the plus on blood type signify?
The plus and minus is referred to it's called the rhesus factor, and that's something different. Okay, but it does affect the transfusibility of it, particularly the mixing of the blood with respect to an infant and the mother. Immunogenic, so that's a good question. I use the term immunogenic. What does the term immunogenic mean? Immunogenic means it stimulates the immune system. So you don't want the immune system tackling something that's critical for you to be alive, in which case blood would be one big thing. You can kill a person by giving them the wrong blood. Yeah. Or cause them a lot of great deal of discomfort. Yes? So the L form of amino acids is by far the most common form. So I'm not asking you to memorize which configuration the amino acids have. Just know that L is the predominant one and the D is much rarer. Yeah. With sugars, we do have that convention, but with amino acids, I'm not going to go through that with you. Okay? Yes? That unusual D-alanine would be in those peptides that are linking together the sugars in the membrane of the bacteria. Okay? So they had what I call cross-links that were holding those polymers of sugars together, and they were tetrapeptides, meaning that they each had 4 amino acids linked together, and that's where you find the d-amino acids. Okay? It's only in bacteria, yeah. Okay. Well, let's turn our attention to glycolysis. So we're now going to turn our attention to metabolism. That is, the chemical reactions going on inside of cells. All right? The biochemical reactions going on inside of cells. I want to start here with the big picture. I'll come back to the pyruvates later. But the big picture of glycolysis is that glycolysis is a metabolic pathway. We call it a pathway because things go from one to the next, 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 to the next. Okay? Just like a road goes from one town to the next town, to the next town, to the next town. That's how you go sequentially through those towns. Okay? This pathway has 10 steps, and you'll notice it has a fork in the road, but it's not really a fork in the road because, as you'll see, everything down below here, they're all identical. Okay? So I'll talk about that when I get down there. But this is a pathway that has 10 steps. This pathway involves the breakdown of glucose. It is probably the most abundant metabolic pathway on the face of the earth. Only other possible exception might be the citric acid cycle. Okay? So it's found in virtually every cell on the face of the earth. Glucose is one of the most common currencies. That is something that's exchanged between different organisms, between different cells, as a way of having metabolic energy. The reason cells are using this pathway is that they're trying to generate energy to make ATP, and ATP is what they use to, as of course, move, to think, to synthesize things, etc. All right. So this is the big picture. I'm going to show you a couple things on the big picture, and then I'm going to show you each of the individual reactions. I'm not going to ask you to memorize the structures here. Okay? The only structures you should know would be ones that I would automatically think you knew from memorizing glucose and fructose. For example, there's glucose right there. I've already asked you to memorize glucose. Glucose 6-phosphate. Well, if you know glucose 6-phosphate because on position number 6, what do? You put on a phosphate, right? Fructose 6-phosphate. Fructose 1,6-bisphosphate. All right? Those are all ones that you're going to know just by knowing glucose or fructose. Okay? Now, the others down here, I'm not going to hold you responsible for knowing. All right. Now, as we look at this pathway, we see a couple of things going on. First of all, we notice that to get the pathway started, we actually have to burn ATP. We have to put energy in to make this pathway start. That's something that's unusual for a pathway that generates energy. One of the things that we see with glycolysis is that it's an unusual pathway in several respects. That's one of the ways in which it's unusual. We'll also see that the way that this pathway is controlled is also unusual. Okay? Now, we have 2 steps that put in ATP. We have 2 steps, or actually 4 steps, that generate ATP. 2 identical here, 2 identical here. So whether you call that 2 or you call that 4 is up to you. That means that 2 ATPs go in, 4 ATPs come out. We have a net production of ATP of 2 ATPs from this pathway. Now that's not the only thing that we get out of this pathway, though. What else do we get? we get electrons, we get reduced electron carriers. We get 2 NADHs. NADH is produced by oxidation. That is, it gains electrons from something which is becoming oxidized, and NAD is becoming reduced to make NADH. We get 2 NADHs, and those will be useful later for making ATP. It's not made here, but it's made later. Okay? And the other things that we get out of the pathway are we get the products down here of 2 pyruvates. 2 pyruvates. Those pyruvates are used in other pathways, and we will see how that happens when we talk about the citric acid cycle. Okay. Now, another thing that I want you to think about with respect to metabolic pathways is that, again, metabolic pathways are like highways. We can think of a highway as saying, well, here is the Corvallis-Eugene highway, okay, to talk about Highway 99, for example, all right? Well, that highway is part of a bigger highway system. 99 goes all the way south. It goes all the way up to Portland, right? And so we've defined a part of it by saying Corvallis to Eugene, that's the Corvallis-Eugene highway. But in fact, it's arbitrary what we decided. We could have said the Corvallis to, I don't know, Salem highway, right? So metabolic pathways are the same way. They're embedded in something else. Something made glucose. Pyruvates are used in other places. So what I define as the Corvallis-Eugene pathway, in this case I'm calling it glycolysis, is only part of a giant metabolic picture. It's part of a giant metabolic picture. So I want you to keep that in mind, because it's very easy to think about, well, this is isolated from everything else in the cell, and it's not. This pathway occurs in the cytoplasm of the cell. Occurs in the cytoplasm, and in the cytoplasm there's hundreds of thousands of reactions going on simultaneously. So we can look at this and say, well, this is going on in this pathway, but in fact we might have molecules of dihydroxyacetone phosphate that are being produced by another pathway that now are available here. We might see another pathway taking away 3-phosphoglycerate and using it for something else. So we're biased in what we are actually looking at here to say this is this one pathway, and these molecules are confined here because they're not confined here. They're spread throughout the cytoplasm. But what pathways give us is a way of thinking about things, all right, that give us a sort of a definition. Otherwise, we think about thousands of reactions at once, and you don't want to think of thousands of reactions at once. Okay. Well, let's take a look at the steps in the process. I do think you should have an understanding of the names of the enzymes. The reason I do that is because I think the enzymes tell you what's going on in the reactions. Okay? The enzyme names tell you what's going on in the reaction. First enzyme. It's an enzyme called hexokinase. The name tells you what it does. What does it do? Hexo means 6. Kinase means what? Puts a phosphate on. So it puts a phosphate onto 6 carbon sugars. It puts a phosphate onto glucose, and you get glucose 6-phosphate. That's a step that requires ATP energy. Okay? The next enzyme, glucose phosphate isomerase. It's also called phosphoglucoisomerase. You don't need to know that. There's a variety of names for that enzyme. It simply rearranges it so that it goes from glucose 6-phosphate to fructose 6-phosphate. So we've simply had a rearrangement of the molecule, and what's happened is this aldehyde became a ketone. That's what happened. The second one is called glucose phosphate isomerase. Glucose phosphate isomerase. Okay. So we can see again the relevant pieces that are changing here. Now, these first two are pretty straightforward. The most important enzyme that we'll be considering in terms of regulation of the pathway is actually the third enzyme. This is why glycolysis is an unusual pathway. Many pathways are controlled by the first enzyme. Remember we talked about feedback inhibition. All right? Well, hexokinase does help to regulate the pathway. It's the first one, but not by feedback inhibition. Instead, this phosphofructokinase is the most important enzyme for controlling the overall pathway. You can call it PFK, by the way. Just call it PFK. Now, I'll talk about regulation later, but this is a very, very, very interesting and a very, very, very important enzyme. You'll notice that it's using ATP energy again. This is where our second ATP is being burned. And it's converting, well, let's look what the enzyme name says, phosphofructokinase. What does a kinase do? Puts phosphates onto things, right? It puts phosphates onto phosphofructo molecules. There's a phosphofructo molecule right there, meaning that we end up with a phosphofructo molecule that has another phosphate. So now as a product of this reaction, we have fructose 1,6-bisphosphate. And by the way, I'm going to give you some abbreviations you're more than welcome to use if you want, okay? All right. Glucose 6-phosphate, you can call that G6P. Fructose 6-phosphate, you can call that F6P. Fructose 1,6-bisphosphate, you can either call it FBP or F16BP. Okay? Now, this is what's described as the first phase. Different books, sometimes books break it into 3 phases, sometimes books break it into 2 phases. We break it into 2 phases, okay? But some books will say, okay, we finished the first phase, now we're going to go on to the next phase, all right? Well, all right, I just noticed there's another typo on there that should say aldolase. But fructose 1,6-bisphosphate is broken in half in the next reaction. It's broken into 2 3-carbon pieces. One 3-carbon piece is called dihydroxyacetone phosphate, or DHAP. And the other piece is called glyceraldehyde 3-phosphate. You guys saw glyceraldehyde yesterday. You can call that G3P. Okay? So we had 6 carbons here, we have 3 carbons here, and we have 3 carbons here. All right. We've got to wade through these reactions before it'll get interesting, I promise you. It'll get interesting, okay? All right. Now, notice what's going on down here. You notice that there are arrows between this DHAP and the G3P, and the arrows between them are there to tell us that they can be interconverted into each other. They can be interconverted. Well, if we're following glycolysis, what happens is DHAP is converted into G3P. The enzyme that catalyzes this is called triose phosphate isomerase. You can call it TPI. And triose phosphate isomerase is a really interesting enzyme. It's an example of an enzyme I talked about earlier in the term where I said there are some enzymes that are perfect. What do perfect enzymes do? Anybody remember? Everybody bashful? Yeah. That's right. So they're enzymes whose reaction rate is limited only by the speed of diffusion. They have very high affinities for their substrates and very high kcats. That's the 2 things that they have in common. Okay? Triose phosphate isomerase is one of those enzymes. Okay? All right. There's the first half. All right, so let's just look at those. There's glucose going to glucose 6-phosphate. Right? And all that's happened is we've gained a phosphate on the glucose. Here is glucose 6-phosphate going to fructose 6-phosphate, simply a rearrangement. We can see that rearrangement happening there. The phosphate's still on position number 6. Here is phosphofructokinase acting. It's taking fructose 6-phosphate and putting a phosphate on position number 1. So now we have this guy that's got 2 phosphates on it. Okay. And here is, I'll come back to that allosterism in a second. Here's the last reaction I just showed you, the breaking of fructose 1,6-bisphosphate into these 2,3 carbon molecules. Not the last reaction, I guess there's one more. Catalyzed by the enzyme known as aldolase. It's misspelled in that previous figure. Okay. And here's the interconversion, triose phosphate isomerase, of these two guys here. In the glycolysis direction, we're going to be moving this guy to the right. So at this point, we will have 2 molecules of glyceraldehyde 3-phosphate. Let's stop and catch our breath. What's that? You want to stretch? Okay, stretch. Get up and stretch. Stretch your legs. Yes. So on exams, yeah, I'll say in a second. What's that? I'm sorry, I can't hear you. Is PFK a rate-limiting enzyme? It is a, I would describe it as a rate-limiting enzyme, yes. Okay? Okay, so the question was, can you use abbreviations on the exam? The answer is yes. You can always use any abbreviation that I use in class on an exam. Any abbreviation that I use in class on an exam. Okay. And by the way, I can tell you one thing I'm not going to do. All right? I am not going to say, write down glycolysis. I may ask you reactions out of glycolysis, but I'm not going to say, give me the 10 reactions of glycolysis. That's not going to happen. Okay? So students think, oh, I memorized the whole pathway, and he didn't ask me the whole pathway. Well, I may ask you something out of the pathway, but I'm not going to ask you 10 reactions of glycolysis. All right. Second phase. We've just finished with the first half of it. Now we're going to go to the second half of it. All right? The second half is where it's a lot more interesting, just like a football game. Right? Football game's never much fun in the first half. It's the second half where the interest is. All right. At this point, we have 2 of everything on the screen because we now have 2 molecules of glyceraldehyde 3-phosphate thanks to triose phosphate isomerase. You may thank that enzyme for making your life simpler. Instead of having a whole 2 sets of pathways to memorize, now you just have 2 of each one of these, identical. Okay. In the next reaction, this reaction is very important and very interesting. In fact, they're all important, but this one's very interesting in the sense that this is the only oxidation that happens in glycolysis. It's the only oxidation that happens in glycolysis. How do I know that? By virtue of the fact it's the only place in glycolysis where NADH is produced. The only place in glycolysis where NADH is produced. Wherever we see NADH produced, it is always produced as a result of an oxidation of something else. Okay? Now, this reaction, there's a lot of stuff going on here. We're going from this guy up here, which is an aldehyde, down here to this guy, which is an ester. An ester is made by joining something to an acid. So that means that aldehyde became an acid and something got attached to it. The something that got attached to it was a phosphate. Now, the other 2 times we put phosphates onto a molecule in glycolysis, the other 2 times we did that, we had to use ATP. We don't have to use ATP here. The phosphate is coming in and getting put on there. My question to you is, why? How? How is that happening? Any thoughts? Yes? I can't quite hear you. Okay, so is the NAD, the NAD is involved, but not quite in the way in which you said. Okay? The hydrogen, uh-huh. Okay, so you guys are getting into mechanism. I'm trying to keep it simpler than that. So you're both thinking in the right direction. The answer is oxidation produces energy. We were using ATP to put phosphate onto those molecules because it took energy to put phosphates onto those molecules. It also takes energy to put this guy on, but the energy comes from the oxidation reaction that's occurring. Oxidation is providing the energy necessary to put that phosphate on. That's saving an ATP. Because the cell is interested in saving ATP, it doesn't want to burn ATP unless it needs to, because remember, it's running this pathway, so ultimately it can get ATP. Okay? Well, this molecule that's produced, 1,3-bisphosphoglycerate, turns out to be really, really high energy. It's very high energy, and it's high energy because these two phosphates really repel each other, and they're tied together in this little tiny molecule. They want to get away. Okay? Well, they get away in the next step in the reaction catalyzed by phosphoglycerate kinase. Notice that we're producing ATP here. Since there's two of these, we're producing 2 ATPs, and we're producing 2 molecules of 3-phosphoglycerate. Okay? That's happening because those phosphates are very high energy phosphates. That phosphate has enough energy that there's more energy in this molecule than there is in ATP, so it readily transfers its phosphate onto ADP to make ATP. Okay? Very important. Now, the next reaction is one that people don't talk about a lot, and I think it's one of the most interesting and important reactions in all of glycolysis. I hope I convince you of that. All right? It looks like it's a very simple thing. We're going from 3-phosphoglycerate to 2-phosphoglycerate. Basically, we're moving a phosphate from position number 3 to position number 2. Oop, and there's an extra phosphate on there. I just noticed. That should not be there. You stare at something for what seems like months, and then you get in front of class and you realize, oh, there's an error. So that phosphate should not be on there. All right. Now, the point is that that looks like a very simple thing, but it's not a simple thing. All right? The reason it's not a simple thing is because that phosphate is not moving from position 2 to position 3, even though it looks like it is. Instead, what's happening is that there's an intermediate being produced. So I need to tell you about that. You see the name mutase on the end of that enzyme? Mutase enzymes have interesting properties. They involve what appear to be movement of things, but they don't actually move anything. They do a little sleight of hand. How do they do it? Well, let's imagine I've got 3-phosphoglycerate sitting here, and I want to make 2-phosphoglycerate. What the mutase catalyzes is, first, it catalyzes the addition of a phosphate to position number 2. That's the first thing that happens. The first thing that happens is a phosphate gets put onto position 2. That makes a molecule that has a name you've heard of before. What did we just make? 2,3-phosphoglycerate. What does that sound like? 2,3-BPG, right? And where did we hear 2,3-BPG? In hemoglobin. And what did 2,3-BPG do to hemoglobin? It caused it to release oxygen by converting it into the T state, right? And what did I say? Where did we find 2,3-BPG being produced? In actively metabolizing cells, right? It turns out that this intermediate is stable. Meaning it doesn't all go down here to 2-phosphoglycerate. Some of it escapes. And that little bit that escapes goes out and tells the hemoglobin, hey, there's a lot going on here. The more glycolysis is going on, the more 2,3-BPG escapes. And the more escapes, the more the hemoglobin knows that these are cells that are metabolizing things. They need oxygen. Okay? So because of this little quirk of this little odd enzyme that nobody talks about, because of this very quirk, all right, your body is giving oxygen to the cells that need it the most. A pretty cool thing right there. Okay? All right. So we've now made two phospho. Oh, and in the next step, the phosphate at position 3 is taken off. Unlike what that shows right there. But the phosphate at position 3 is taken off, and you're left with 2-phosphoglycerate. Okay. Well, the next step is pretty straightforward. 2-phosphoglycerate is converted. I got to sneeze. I'm allergic to 2-phosphoglycerate. Damn it. You know, when you start to think, I've got to sneeze, and then you don't sneeze, and then I'll say something, all of a sudden I'll just blow snot all over everybody. Okay, I'll try not to do that. Anyway, in the next step, water is removed from this guy to make a double bond. That's catalyzed by an enzyme known as enolase. And enolase is important because what it's creating down here in phosphoenol pyruvate is a very high energy intermediate. A very, very high energy intermediate. We saw yesterday when I showed you the high energy phosphates that PEP, phosphoenol pyruvate, had about twice the delta G0 prime value of ATP. It was about minus 62, and the delta G0 prime for ATP's hydrolysis was minus 31. This guy is loaded with energy. It's loaded with energy. Now, the reason this is important, okay, the reason that this is important is because in the next step we're going to convert ADP into ATP, meaning that this guy can transfer a phosphate to ATP with no problem whatsoever. But it doesn't transfer two. It doesn't make two ATPs. It only makes one ATP. There's some significance of that as well. All right? When we have a lot of excess energy and it's not used in making bonds, what do you suppose happens with that energy? Not stored, no. It comes off as heat. Okay? Any energy that's not used for something comes off as heat. This reaction is an example of what I like to call the Big Bang reaction. It's catalyzed by an enzyme called pyruvate kinase. And it's big enough in that it not only makes ATP, but it generates a lot of heat. You wonder why you get hot when you go running or when you're exercising? Now you know part of the reason why. You're doing a lot of pyruvate kinase reaction and producing a lot of excess heat that you got to do something with, right? Well, you're going to sweat it out. You're going to lose that heat, right? And so forth. And of course, you can generate heat in these other reactions as well, but this guy is generating a lot of heat. Okay? The more you go through glycolysis, the more heat you're going to generate. Okay? Well, we've reached the end. We now have made 4 ATPs, we've made 2 NADHs, and we've made 2 molecules of pyruvate. Yes, sir? Good question. Is this an energy neutral reaction? This is in fact an energy neutral reaction. Yes, it is. Okay. All right. There are 3 reactions that are very energetically favorable. Okay? 3. The first reaction with the hexokinase. Yes, I think you should know this. The third reaction with the PFK. And the last reaction with the pyruvate kinase. Those are very energetically favorable in the forward direction. That means they're very energetically unfavorable in the reverse direction. Yes? It does. So the ATP energy is used partly to make the bond, but it's also partly to drive the reaction forwards. Same thing with the PFK. If the ATP was not used, it would be an energetically unfavorable reaction. So when we look at the reaction, I see you frowning. When we look at the reaction, okay, the reaction is the product of everything, including the hydrolysis of ATP. So the ATP hydrolysis contributes to the energy of the reaction. Okay? Now, those three reactions that I just told you, hexokinase, PFK, pyruvate kinase, those three reactions turn out to be three reactions that regulate glycolysis. Glycolysis is very unusual. It's got three things that regulate it, and one of the things that regulates it is the very last enzyme. The very last enzyme is a regulatory enzyme. That's really odd. Now, there's a reason why. I'm not going to talk about it right this minute, but there's a reason why that happens. Okay? There's one reaction in the pathway that is energetically fairly unfavorable. Which one is it? It is, in fact, this guy right here. This reaction has a delta G0 prime of about positive 20. Positive 20. Does that mean that that reaction doesn't occur? No, that's right, it doesn't. Delta G0 prime does not determine the direction of a reaction. What does it determine? It determines an obstacle we've got to get around to make it happen, right? If I said that this is the reaction that I have and it's got a delta G0 prime of plus 20, how could I make this reaction go forwards? I can't change the temperature. So in a living cell, I don't have that option. What's that? More reactants. So if I dump a whole bunch of this stuff in there, then what's going to happen? More reactants is going to make that denominator greater. It's going to make that log term become more negative. And if I put enough reactants in there, I will make it a negative 20 or greater, right? There's something else I can do. What else can I do? I took away. It's actually easy, guys. All right? There's one other thing I can do. What's that? Take away products. If I remove products, all right, it has exactly the same effect because I'm making that numerator be smaller, the denominator larger, and I'm making that log term be more and more negative. In fact, as we will see, cells do both of those. Cells have to play with concentrations to make pathways go the direction that they want because that's the only thing that cells have control over. They can't change the delta G0 prime. That's fixed for a reaction. But they can change the amounts of reactants and products. Okay? All right. Now, I'll show you a cool trick. All right? Let's go to the big picture. Here's the problem reaction right here. Splitting. Okay? There's the problem reaction. We said that what you can do is you can increase the amount of reactants and you can take away the products, right? I'm going to tell you a way that your body does this, and it's pretty darn cool. Let's imagine, if you will, that you are sitting in this room and the fire alarm goes off. The fire alarm goes off, your adrenaline starts flowing. Your adrenaline causes your muscles to release glucose from glycogen. Your blood glucose levels go up. The concentration of glucose in your muscles, where you need them, goes up. Okay, are you with me? Glucose goes up. What happens to the concentration of glucose 6-phosphate? It's going to go up. What happens to the concentration of fructose 6-phosphate? It's going to go up. What happens to the concentration of fructose 1,6-phosphate? It's going to go up. But wait. I've got a barrier here. Okay? If all I do is increase this and I don't decrease this, then I've got a bit of a problem. Okay? Well, I can make fructose 1,6-bisphosphate in abundance based on what I just told you, but it sure would be nice if I could use this to help me to jumpstart glycolysis. And your cells have this built into them. Really, really cool. All right? Why? Okay. Remember I said pyruvate kinase is a regulatory enzyme. It helps control glycolysis. Pyruvate kinase is activated by F16BP. It's activated by it. Now, I'm going to step you through what that means. All right? What we've done so far is we've dumped glucose, we've made glucose 6-phosphate, we've made fructose 6-phosphate, we've made a ton of fructose 1,6-bisphosphate, but we've got this energy hump to get over. Fructose 1,6-bisphosphate is accumulating, and when it accumulates, some of it goes down here to pyruvate kinase, and what does it do? It turns it on. So pyruvate kinase starts converting PEP into pyruvate, which decreases the concentration of PEP, which decreases the concentration of 2-phosphoglycerate, which decreases the concentration of 3-phosphoglycerate. You get the picture. Which decreases ultimately the concentration of these two guys, and we've just pulled the whole process forward. Now that's cool. That's pretty darn cool. It's built into your cells. You didn't even know it was there, and it happens every time you get up and you're scared. That's pretty cool. Yeah.
What exactly was activated by?
So pyruvate kinase down here, this enzyme is activated by this molecule right here. Now this is one of the very few cases in all of metabolism where we have what I call feed-forward activation. I've talked about feedback inhibition, but here's feed-forward activation. This guy is telling the enzyme ahead of it, get your butt in gear, because we're going to need to start glycolysis like crazy. Pretty cool. Feed-forward activation. Okay. Should we have a song? Okay. This is to the tune of a few of my favorite things. Aldehyde sugars are always aldoses, and if there's a ketone, we call them ketoses. Some will form structures in circular rings. Saccharides do some incredible things. Onto a glucose, we add a P to it. ATP energy ought to renew it. Quick rearranging creates F6P without requiring input energy. At a high rate, add a phosphate with PFK. F16BP is made up this way so we can run and play. Da, 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 da. Aldolase breaks it and then it releases DHAP and a few G3 pieces. These both turn into 1,3-BPG, adding electrons onto NAD. Phosphate plus ADP makes ATP, while giving cells what they need, energy, making triphosphates a situation of substrate level phosphorylation. 3-BPG, 2-BPG, lose a water. PEP gets a high energy state just to make pyruvate. Da, 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 da. So all the glucose gets broken and bent. If there's no oxygen, cells must ferment. Pyruvate, lactate, our cells hit the wall. Some lucky yeast get to make ethanol. This is the end of your glucose's song, unless you goof up and get it all wrong. Break it, don't make it to yield ATP. You'll save your cells from futility. All right.